Well, thank you very much for your presence. We have with us today for a spotlight interview, Doctors Michael Van Dyke and John Barrows, both researchers at Kennesaw University in the United States. So Michael has had a lifetime interest in studying ligand nucleic acid interactions and associated methods development. His career spans over 40 years of small molecule DNA interactions, general eukaryotic transcription mechanisms, immobilized methyl affinity chromatography, G*G multiplex nucleic acids, combinatorial selection methods, lysine deacetylase regulation by inflammation, and ribosome homeostasis under nutritional stress conditions. His most recent work includes a reverse genetic approach towards understanding the potential biological functions of putative transcriptional regulatory proteins in the model extremophile organism Thermus thermophilus, examining refractory cleavage sequences for type 2S restriction endonucleases, and developing methods for the study of ligand nucleic acid interactions at primary undergraduate institutions. John Barrows is currently a National Science Foundation postdoctoral research fellow in biology at Kennesaw State University in the United States and studies transcription factors in thermophilic bacteria. John received his PhD in biomedical sciences from the Medical University of South Carolina in 2021, where he investigated mechanisms of transcription and DNA repair. He received his bachelor's degree in biochemistry from Clemson University in 2016. And today, we will be discussing the PLOS ONE Lab Protocol paper entitled Biolayer Interferometry for DNA Protein Interactions and the Associated Protocol in Protocols.io. Thank you so much, both of you, for being here.
Thank you.
Thank you.
So if we can just kick off by you briefly explaining what this protocol is about, how to use it, and why it is relevant.
I might start this. So biolayer interferometry is a technique that uses a change in an interference pattern to ascertain whether a ligand, usually it's a macromolecule, is associating with another one on a substrate. So there's a machine, a biolayer interferometer, I think the octet systems. Is that correct, John?
Yep.
Yep, he agrees. It's sort of the primary way people go nowadays. And what was absent a few years back, this is like about 2016, is a good protocol for this. We just didn't have one. So it was a very useful technique earlier on for us. We're at a primarily undergraduate institution. And for those who aren't familiar with these, it's teaching is our main focus. And we have a lot of very junior students here. So it's not your usual PhDs, postdocs, et cetera, institution. Also, we have no radioactive license. So we had to have technologies that allow us to do things in the absence of that. And BLI is a beautiful example of that because no radioactivity is needed, yet you can get some real-time kinetic measurements on things associating or dissociating from one another, including proteins and nucleic acids. John?
Yeah, no, that's a good summary. I will say we're probably going to use the terminology BLI quite a bit, which is just an acronym for biolayer interferometry. Yeah, a lot of stuff in the literature for protocols regarding BLI were usually for protein-protein interactions. So something that we study and Michael studied before I got here is just DNA protein interactions, and using this very powerful technique, which has been used for protein-protein interactions for kind of something we were interested in, has been very helpful. And several projects have used this kind of experimentation for certain things like validation.
So if I understood correctly, the motivations for creating or optimizing this protocol is to make it work in a facility that doesn't have radioactivity. Is that correct?
That was the original motivation for it. It was also a device that we have here. So if you were, let's say, down the road at Georgia Tech or Emory, they would have things that are, or even Georgia State, have a device that's called surface plasmon resonance, or SPR. It's an alternative technology. It doesn't work with light. It works with like electric fields that might exist on the surface. And also a very powerful technique for doing ligand, nucleic acids, protein, DNA, protein, et cetera, many different things. The problem is we do have an SPR here at Kennesaw, and it is crazy expensive to run. Like one experiment's like $400. That isn't where we come from. So when you do BLI type experiments, you're running more on the $4, $10 scale. So it's logs different for one another. And again, doing things with undergrads, this works much better here. But it's definitely translatable to industry, to better institutions, et cetera.
So it's a more affordable and safer methodology.
And it gives quite good values. So perhaps we're not at the top tier gold standards that you might get in some other techniques, but it does give very useful numbers. I mean, we're trying to ascertain, does it bind tightly? Does this mutant bind better than that one? You can get all that kind of data easily from this.
So you invested all this time into optimizing this protocol that gives you a very good compromise or ratio of quality to safety and cost.
Right.
But what was the motivation to upload it and make it public in the platform after you already devoted all this time into the optimization itself?
Well, I've been interested in actually doing protocols for a long time. I mean, even like graduate school, a lot of what I've done has sort of been, you develop a new technique, new technology, apply a machine in a different fashion, and then it opens whole doors for what you do. Maybe in the past that was just part of a paper, but since I've come here, I now appreciate more the educational aspects and disseminating this information broadly. I didn't know about protocols.io until, God, I think it was like 2017. We had sent a paper into PLOS ONE, and the editor there said, hey, have you ever heard of protocols.io? You know, you ought to submit this as a protocol there. And that's how we got started.
So actually your initial approach then was to publish it as a methodological paper in PLOS ONE.
No. It was, so when you write a paper, you can put in your experimental. Our experimental is very long and very detailed. She said, that's not really the right thing to do. Why don't you put this in protocols.io and then reference it instead? So that's how we got started.
But and then what was the motivation for having peer-reviewed independently as a lab protocol paper?
I looked that up because it was, I think her name was Emma. She's at protocols.io. What is her
Emma Ganley, yes.
Yes, that's her. She had contacted us for a protocol. We had done one, actually, John, this is right before you were here, is the REPA one, Restriction Endonuclease Protection Assay. We had just put that out, and she said, oh, you know, we have this new thing going on. I think it was in 2017, where they were going to also publish it in PLOS ONE. And unfortunately, the student who was involved in that had moved on. So I said, I got this new postdoc, and he's really interested in working on this BLI technique. Maybe we could do that instead. And she said, oh, that's fine.
I see. So you got an invitation to submit a different protocol, but
Right.
It was this one because it's the previous one. The hands-on person had already moved on.
Exactly. And I mean, I can say what I know about this protocol. I was doing it back in the beginning, but actually it has evolved considerably under John's hands, and he's now the expert. When people call, I just refer to him.
And how the feedback that you received from the peer reviewers shaped the final version of this protocol?
That I will defer to John, because I looked, John, to the old responses to reviewers and such, but I think you're a better person to talk about that.
Sure. Yeah, it was basically cleaning up, adding a little more information, things that we probably took for granted when we were writing up the protocol, just certain buffer conditions, temperature conditions, a little more information of what's already out in the literature and what can you say of how this is greatly advancing the field. That's kind of the responses that we got from the peer review, which was great. The technique seemed very sound. There was no issues there. And I think that's another reason why we wanted to go to a peer review process through this, because having other experts look at how you're doing things, it's just more eyes on the technique. And you might be so routine in doing something you don't realize if you're making a slight error here or there. So it was good. We wanted that added expertise through what the protocols.io and PLOS ONE were offering.
And now I would like us to dig a little bit more into the development of an optimization of the protocol itself. And I don't know if you could share with us what things during the development and optimization you tried but didn't work and didn't make it to the final version of the protocol. Were there things that you thought, oh, this should work, and at the end you had to change it?
I think the biggest thing for this assay, which we talk about in the expected results, we kind of have a paragraph devoted to it, is every protein is going to act a little bit differently. So the buffer that you use in this protocol is very important. Proteins that have a high PI and might just kind of non-specifically associate with the backbone of DNA will give you artificial curves mimicking association to any piece of DNA. So we also reiterated the need for a negative control in this experiment. You need to add some DNA that you think it should not bind, and you should hopefully see no evidence of binding. But if you do see evidence, that's likely a non-specific interaction, and changing the buffer, adding a little more ionic strength usually just knocks that down. And then you can see a very positive result to your DNA of interest. So that I think for our kind of troubleshooting with BLI, that's the major thing, is make sure that you have a condition where your negative control is flat, and then you can start screening some DNAs of interest.
And in addition to the buffer, are there any other steps that are critical for the run of the protocol?
Well, one thing that I want to stress with regard to it is, again, we're using materials that we get from other people, like the probes themselves. And you got to be careful what batch you use. I mean, we have one batch that it just gives you beautiful data, and then the next batch will give you like half the amplitude. Is that correct, John?
Sure, but it's just don't mix and match different batches.
But are we talking here about the same protein, just different batches of purification, or?
No. Different batches of probes. So the manufacturer, and it used to be Paul, and they've gone through a number of different distributors here in the United States, but I don't know who's supplying them. It's not been 100% uniform, in my opinion. Oh, hey, you might want to strike this from the with editing.
And so that's one of the troubleshoots that you have to do.
Well, yeah, and John was absolutely spot on. You need to do control. And for us, it's great as an educational thing for students is, you know, if you get this wonderful looking data, but your controls look the same, you're not really learning anything. So the importance of your negative controls, if you had a positive control, that would also be useful, but just having your controls there before you start telling us numbers as far as this is the K on, K off, et cetera.
And since the time of publication of the protocol and the paper, have you done any further optimization of the protocol, or have you taken it to new or different avenues?
In terms of optimization, no, we've kept things very similar. However, once again, each protein, the buffer is going to be a little different. So we just, I believe it was the CSOR. We just published a paper where we used this technique, but it needed just about double the ionic strength that we normally use. And that's once again to just knock down, there was some minor binding to a negative control DNA sequence. So just adding a little more salt totally got rid of that while keeping the, what we found its consensus DNA sequence to be totally fine. It didn't affect that sequence at all.
And how do you get to the right ionic strength? Is this trial and error, or?
Typically, yeah, it's trial and error. We have the Octet 8 system, so you can run 8 kind of different experiments in one run of the machine. So it's really nice because you can take 8 different concentrations of salt with a negative control sequence and just see where does it give you very little binding, and then take that and then screen DNAs of interest.
Very interesting. And I would also like us to discuss what has been the impact of getting this paper peer-reviewed and published as an index article for both of your careers.
Oh, well, to begin with, we're both recipients of NSF funding, the National Science Foundation here in the States. And I mean, they have their straight scientific impact and such, but they also have broader impacts. And so dissemination of knowledge is, you know, to a diverse community is very important to them. And they really like the what we've been doing with regard to publishing protocols, whether at Protocols.io or through PLOS ONE. It's something that my program officer has been quite pleased with. And if I can keep him happy, that's a good thing.
Yeah, I
and
think
for you, John, as an early career scientist?
Yeah, I think it looks great on a CV. It shows that you can take something and kind of lay it out and describe exactly what you did and how to troubleshoot this type of experiment. And then in terms of not really career, but just helping the scientific community, I know I've personally had several people reach out and kind of say like, hey, I'm trying to do the same thing. And I've helped them walk through the experiment and the output and how to analyze data and things like that. And I've also been tapped as a reviewer for just a BLI experiment on a paper. They wanted someone who was familiar with the technique. So they reached out to me and said, hey, can you just look at this? And does this check all the boxes? Did they do this correctly? So that's been cool to see that it is getting readership and people are interested in it.
That's fantastic. I don't want to take much more of your time. So before we wrap up, I just wanted to know if there's anything that you would like to mention that we haven't touched during our conversation.
Not for me. We're happy that we're able to participate in this fashion. So it's all been good.
Yeah, thank you for reaching out. And I will just say one other cool aspect about this kind of experiment, which I think appeals to kind of the undergraduate mentality, is you can see results in real time. So you have a screen that's showing you exactly what's happening in the machine. And for students, that's really exciting, because other experiments, you know, it just takes time and they don't like waiting, and they just sit around and they're just waiting for the results. So this one's cool because you set it up and then you can just sit there and you watch it unfold right in front of you. So
it's a protocol that is suited both for research, but also very good for teaching.
I would say
Oh,
so.
yes,
Yeah.
definitely.
Yeah, well, very interesting. Thank you so much for your time. Thank you for your insights. And I wish you a happy rest of your day.
Thank you
very much.